TK, back to you guys, back to YouTube. All right. So guys, yep, took a little break. Back now. Identification of formed elements. All right. So blood cell model. I showed you what that was. Okay. You guys were supposed to look at it, find the leukocytes that you're supposed to. Okay. It had a key attached to it. It wasn't very hard. You just had to match up the numbers on the model to what the key said. Give you a little introduction of roughly what they would look like on the model. Okay. But we also looked at slides, and we also have some pictures down below, all right, which we're going to talk about in a second. But first, I want to work on this table with you guys, okay? There's a lot of stuff in here you got to know. So table 1.3, if you're looking for a good strategy for studying, if you guys know everything in table 1.3 about all the formed elements, you guys will be very, very solid with answering any question we throw at you, asking about these formed elements, okay? So if you get this down, you'll be perfect, okay? Well, let's go through this together, all right? So let's start with classification, okay? Specifically for the leukocytes. So erythrocytes, they don't have their classification. Platelets don't either. But all the leukocytes, remember, they do. Are they going to be agranulocytes, or are they going to be granulocytes? Strictly, okay? So just to simplify this, because of how small the boxes is here, I'm just going to write an A or a G, respectively, okay? And if we're going to think about this, remember, we gave you guys the mnemonic here, okay? So we have Ben-G. All right, this will answer it for you. And we have mol for our last one, okay? Where the G and the A specify on what each one is, okay? So let's talk about the granulocytes, okay? BEN, match it up here. B, basophil, so this one's going to be a granulocyte. E, eosinophil, granulocyte. N, neutrophil, granulocyte. Easy. All right, those three right out of the way. That leaves the last ones then, okay? So A, lymphocyte, M, monocyte, A, granulocyte, okay? So that's it. Very, very straightforward. If you know these mnemonics, you guys will be very, 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 very fine. It's going to be very, very easy for that question, okay? Ben-G, mol, boom, easy. All right, appearance. So how do these guys look, okay? Let me see if I can zoom in here and add a better text box for this. So shrink down my lettering here. Good. So what do erythrocytes look like, okay? So the answers for this, remember, are going to be found on the following pages. Talks about the characteristics, but let's fill it into the table, okay? So what do these guys look like? Remember, these are the most abundant. They're everywhere. They look like these red, pink, concave shaped discs, okay? Shrink that up here. Beautiful. So that's what they look like, okay? And if you're missing this, oh my goodness, there's no way you can be missing this because they're everywhere, okay? You can't be missing this. All right, platelets now. So I'm just gonna, perfect, get that down there. How do these things look? So remember, I describe these things as looking like little polka dots, okay? So let's scroll down just to see these guys quick. All these very, very small little purple polka dots here on the stained image are the platelets, okay? So very, very numerous, not as much as red blood cells, but they're there, okay? They're in higher numbers compared to the leukocytes. So those little, very tiny polka dots are platelets, okay? So how am I going to describe these things? Very small cell fragments is how I will describe this, okay? Perfect. All right, now neutrophils, okay? So if we scroll down, you can look at the pictures here, okay? It's about a 3 to 5 lobe nucleus in an S or a C shape. You can have some color in there too, okay? So 3 to 5 lobe S or C shaped nucleus with, you could say the color here, with pinkish, remember this is a granulocyte, so you can say pinkish granules, all right? Bundle this guy up, good. All right. Eosinophil. I'll talk about my little, tip and trick here, how you guys memorize this, but this one has a 2-lobed nucleus, and we'll go over that in a second, okay? 2-lobed nucleus with a red/pink color. All right, because eosin means red in Greek as well, okay? Get that guy there. Basophil, all right? B, all right, basophil here. This one is very, very dark. Okay, has very, very dark granules when stained, so it looks very, very dark, okay? So how am I going to describe this one? So remember, you can't really see the nucleus, but it's there, okay? So nucleus hidden behind, gosh, my handwriting, man, behind the granules, okay? So hidden behind the granules here, very, very dark That's all I really got to say there, okay? Because when it's stained, it turns that very, very dark color, all right? Very, very dark purple. Lymphocyte looks very similar to a basophil, all right? Remember, lymphocytes don't have any granules, so it doesn't kind of have like that grainy, looking appearance to it. Okay, it looks very smooth, all right? So what this will have is either a small, medium, or large shaped nucleus. Doesn't have any lobes on it or anything, okay? It will cover the majority of the cell. So it won't cover all of it. It won't look like the basophil where it looks like the basophil is covering all of the cell, okay? But it's going to have a small, medium, or large shaped nucleus that will cover the majority of the cell. We'll take a look at that picture, okay? Monocyte, what is this one? So this is a very large cell with a C-shaped, all right, and this is a singular lobe, or one, I can't even say. A C-shaped, I will just say she-shaped nucleus. All right. So very, very straightforward. All right. So if it has a C shape, it doesn't look like it has any lobes to it. All right, it's a monocyte. All right. So formation process, okay? Remember, formation process here, all formed from hemopoietic stem cells, all right? But if it's going to be a poiesis, we're going to say erythropoiesis. All right, that's the process there. Platelets is thrombopoiesis. Neutrophils, and all the ones going down here, okay? They're going to be leukopoiesis, all right? And we'll just copy. I can't copy and paste. Just know that this is going to go down for all of them, just like that. Applies to all of them, for all the white blood cells, okay? All right, relative abundance, okay? Remember, erythrocytes at the top, number 1. Platelets, number 2. What's our mnemonic, remember? Never let monsters eat. Biology in that order, okay? So 1, 2, so this one will be 3, 4, 5, 6, 7. So let's fill it in. Neutrophils. 3, 4, 5, and then 6, 7. All right, easy. All right, relative abundance, easy. Never let monsters eat biology, but erythrocytes and platelets first and second, respectively. All right. Function, okay. What are the functions here? So red blood cells. So the responsibility of this is to transport oxygen. That's the big one, okay? Transport oxygen, and you could also say carbon dioxide too. We're trying to expel the oxygen that our tissues used up. It transforms into carbon dioxide, which we'll talk about in the respiratory system unit in a few weeks. Platelets, what are these guys responsible for? Remember, they're responsible for hemostasis. So remember, clotting our blood and everything. Vasoconstriction as well. So this is kind of like the squeezing, kind of like putting if you have like a garden hose, like squeezing it nice and tight shut. So it makes it harder for the blood to move through, makes it harder for the water to move through. And coagulation. All right. Another fancy word for clotting. All right. Neutrophils. So what are these guys responsible for? Okay. I always like to think of all these things when it comes to the functions in terms of military terms, okay? So I always thought of the neutrophils as being like the infantry units, okay? These are our first responders. They always go in, first responders, and their main job is to kill bacteria, all right? The big job, going in to kill the bacteria, okay? So, up next, eosinophil. What is this guy? So I like to think of this guy, all right, as the heavy guns, all right? Big time guns. So this guy's main job is to kill the parasites, okay? So kill parasites. You could also say here phagocytize allergens. So it's basically just kind of cells eating, all right? So eating the allergens, all right? Allergens. And then also to activate basophils. And to activate basophils, all right? Scrunch this guy down. All right, basophils. So what do I always think about? So with basophils, I like to think of these guys as basically being like the alarm, okay? So whenever there's a breach, right? Breach in the base, breach in the system, breach in our immune system especially, the basophils are the ones to sound the alarm. And they do that by releasing histamine and heparin. which are some really, really important things that we use, chemical mediators. Histamine is really responsible for inflammation along with heparin. Inflammation attracts other white blood cells to the area, so they're the alarm. Once they release that histamine, they send alarm and word to the other white blood cells to come to the area, help take care of whatever's going on there, okay? So I like to think of these guys as being like an alarm system. All right, so we have lymphocytes here, okay? So lymphocytes, they can be broken down into T cells and B cells. That was mentioned on the last page here. So the T cells and the B cells. T cells are the ones that recognize and destroy infected cells. So these guys are kind of like our commandos, our special forces, okay? So we'll say here, so they help kill infected cells, all right? And then in particular, the B cells, all right? I like to think of these guys as kind of being like our, I like to think of the B cells as kind of being like our intelligence officers. So they help create antibodies, all right? So for next time, when we have an infection, they can recognize it and attack it immediately, okay? So they create antibodies to recognize infection for the next time, just in case we get another infection. They come prepared, all right? All right, and then we have monocytes, our last one down here. So what are these guys? These guys are basically like our cleanup crew for our white blood cells. So what they help with is for phagocytizing, all right? So phagocytize foreign and dead cells. All right, that's what these guys are kind of responsible for. So our cleanup crew, so the military cleanup crew. So after war goes in, right? After all of our other white blood cells, after we drop bombs, we try and destroy all the infectious material. The monocytes are going to come in and help clean up all those dead foreign cells, help clean up the area to get it ready for healing, essentially. All right, so that's what the monocytes are responsible for. Okay. All right, table here. If you guys can get this down, get all this stuff down here, you guys will be golden. All right, when we throw you a question asking about this stuff, whether it be the appearance, whether that be its formation process, whether that be its function, etc. You guys will be just fine if you know all this stuff here. Okay. Okay. So, talked about these pictures a little bit. Let's look in some of these in particular. I'll kind of run you through my tips and tricks for finding some of these. If we were to show you guys a picture of some of these things, how do we know what we're looking at? So remember, erythrocytes, really, really hard to miss. It's almost impossible to miss because every single stain that you see, it's just going to be littered with this stuff, okay? They don't have a nucleus. They look like concave discs. It kind of looks like they're kind of hollow in the middle. That's just because the concavity on the other side is so large that it looks like it's transparent all the way through, okay? But I promise it's 3D. It's not a donut. There are thin centers, as I said, that appear almost hollow. That's because of the concavity. Platelets. They look like small cell fragments, those little polka dots that kind of sit in between all of the red blood cells. They can be single or they can emerge in small clusters. So this would be like a single one here, a little cluster right here. My cursor is pointing, little cluster, okay? All right, neutrophils, all right? So this is where things can get a little wonky because we have a whole bunch of cells. Some of them look the same, some of them don't. What's going on? Neutrophils, remember, this is the most abundant of the white blood cell, okay? So because of that, I'm going to add a little star here. So if this is the most abundant white blood cell, sometimes you got to just say, when in doubt, if it's the most abundant, that's going to be my guess. But my way of knowing what this is in particular is most abundant in terms of how many lobes it has, okay? Lobes and abundant white blood cell, too, okay? So as we're going to see as we look through all these, this one has the most lobes out of all of them, okay? They appear in this S or C shape, fine light pink purple granules in the cytoplasm and pale cytoplasm. You can count all of them here, right? 1, 2, 3, 4, 1, 2, 3, 4, 5, 1, 2, 3, 4. So 3 to 5 nucleus or lobe nuclei, all right? Eosinophils, all right? Next one here, okay? Large 2-lobed nucleus, large pink red granules. It's in the name here once again, okay? I told you guys this. So remember eosin, okay? So what eosin means in Greek is like a red or pink color, okay? So eosin, red/pink in Greek, okay? But remember my special way of memorizing this, okay? In the Christian religion, it is a sin, remember, to have more than one significant other, okay? So because my friend here has 2 lobes, okay, he has 2 significant others. That's a sin. It's in the name, all right? So it's a sin. You can't have that. So remember, Christian religion equals a sin to have more than 1 sig other. So since he has 2, it is a sin because of that. Hence why it's called an eosinophil, all right? If you want a little easier way to memorize this and what you're looking at, okay? So little tips and tricks right there for this guy. All right, basophil, all right? So it has a grainy looking appearance to it, okay? You can kind of see all the purple, dark purple granules. Hides the nucleus, okay? So it usually has a U or an S shape, okay? But since it's hidden behind all these granules, it's almost impossible to see. Remember my tip and trick for this guy, okay? I told you guys when I was a kid, I hated my basement because it was dark. Okay, I was scared of the dark when I was a kid. All right, so when I see BAS in the name, I'm thinking basement. Okay, and when I'm thinking basement, I'm thinking dark. All right. Now it looks similar to lymphocyte. We'll talk about how we distinguish these two, but this is how I look at it. So basophil, dark, and it basically covers up the entire. All right, dark, entire cell. All right. Because almost the entire cell, basically the entire cell looks dark and scary. You can't see it, okay? That's my tip here. Lymphocyte, remember I said looks very, very similar, okay? Can either come in small, medium, or large sizes, all right? It has a large round nucleus, but remember it covers most of the cell. All right, covers most of the cell. In contrast, basophil, it basically covers all the cell. We look at this thing, it covers all of the cell. We can see these little dimples here. We can see a little bit of that cytoplasm on the inside of the cell here. So the nucleus covers most of the cell, okay? That's how we recognize this guy, all right? And I also said that the L stands for a large, okay? Because it has a large nucleus. Even though it doesn't cover all of the cell, it's still very large compared to all the others, okay? So it's very, very large. And our last one down here, monocyte. Remember, this one was in the name, okay? Mono. So it has a large horseshoe or, C, U-shaped nucleus. See it here, very, very straightforward, horse-shaped, okay? This one's easy, okay? Because mono, all right? Mono in Greek, what does this mean? This means 1. That's because this guy only has 1 nucleus, all right? There's no extra lobes to it, it's just 1 nucleus, 1 lobe nucleus, if you want to be more specific, okay? And that's how you determine it, all right? Large horseshoe-shaped nucleus, pale cytoplasm, largest of the leukocytes too. Another way to look at it, okay? All right, guys. Well, hey, those are the tips and tricks down here. So remember, we looked at the blood sides. Obviously, we aren't going to be doing that on our quiz coming up, our celebration next week. But just another way for you guys to connect what you're seeing on the paper to what you should be seeing in the real world if you decide to go into some sort of a hematology course, or if you decide to go and look at immunology, look at white blood cells. This is a good little stepping stone for you getting to that pathway if you decide to do it. Okay. Okay. Stop here. Blood types after this. Talk about that. Let's scroll up here. Let's get our learning objectives highlighted here. So we talked about all the formed elements on the slides, the images, the blood cell model. We named their functions. All right. And then we weren't really able to do this, but if you guys looked on the slides, you know what a leukemia or a sickle cell anemia would look like. Because remember, leukemia has all those kind of crappy looking white blood cells. You really can't determine, what they are. Like they don't really have a distinctive like a neutrophil, lymphocytes, basophil, shape to them or appearance. They just kind of look like these dark purple, just just kind of sprinkled all over the stain. And then remember sickle cell anemia, guys. Perfect world should have that sickle cell shape, so that crescent shape to it. But remember, it can be. I saw a 4-leaf clover out yesterday on one of the microscopes. I've seen ovals. I've seen rectangles. You have all these very random shapes. So that's what you're going to see with sickle cell anemia stains. Okay. So we have 3 more learning objectives to go. All right. We're going to talk about the blood groups very, very quickly because I know you guys have been over this many times. We're going to interpret the results from agglutination reactions to determine someone's blood type and then determine blood type compatibility. All right. So we'll do that in our next video.